to today's Life Sciences lesson. My name is Vedika Maraj, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Cybono Discovery Center. Please remember that you can WhatsApp us on our WhatsApp number. It is 079 454 9535. Please remember that you cannot send photos or voice notes. You need to type in your questions, and then we will answer you as best we can. I'll give you the number again. It's 079 454 9535. You'll also find the number at the bottom of your screen. Right, so today we're going to do the DNA code of life. We are going to do the first part, which is going into the DNA and RNA structures, DNA replication and the significance thereof, and then DNA profiling. But let's just go to the previous slide. And this topic, if you look at your examination guideline, it gives you an indication of how deep you need to go into the topic when you are studying the topic, right? The next slide I'll give you more detail. But it also gives an indication of how many marks are allocated to this topic. So this particular topic can count 27 marks in your final exam, as well as your preparatory exams in September. So this shows you the possible questions that they can ask you at the end of the year. Those are terminology, interpretation of diagrams, tabulation questions, description questions, and there can also be some calculations. Okay, so you must be able to answer these type of questions. And the best way to approach this topic and every topic in life sciences is to go through past question papers. That is where you will see the kind of questions that they ask, the type of questions that they ask, and also you can have marking guidelines that can assist you with how to answer the questions. So going to a little bit more detail, this is taken partly from the examination guideline, but you should look at your examination guideline very clearly for every topic in life sciences, because it's going to give you an indication of what it is that you need to study. It's basically your scope. So the DNA and RNA, the structure, what do you need to know about it? You need to know the structure, the location and the functions of DNA and RNA. DNA forms genes which carry hereditary information. DNA contains coded information for protein synthesis, and the RNA plays a role in protein synthesis. So we won't be doing protein synthesis today, but we will be doing it next week, so please stay tuned for that. Then we have DNA replication. The DNA makes a copy of itself so that it can be shared between the resulting daughter cells during cell division. Cell division, we have mitosis and we have meiosis. Mitosis you did in grade 10. Okay, that is one cell division that you know. It's in charge of, it basically causes growth and repair. And then we're going to have meiosis, which is the formation of our sex cells, our gametes, which we're going to do later on this term. Then we've got DNA profiling, the uses and interpretation of DNA profiles. That's what you need to know. Each person has a unique DNA profile. A profile is represented as an arrangement of black bars representing DNA fragments of a person. Okay, and then the last part is protein synthesis. So, firstly, what we need to understand before we go into this even is that grade 10 work is very important for grade 12, right? So the foundation that you had in grade 10 is going to help you with your understanding of life sciences in grade 12. So one of the important things is the structure of the nucleus. Now, just to remind you that remember that our body, we have cells, and in the cell we have a structure which is called the nucleus. So in order for us to understand DNA, we need to remember where the nucleus is and what the nucleus looks like. So I have a diagram over here, and it shows us that in the center we have a nucleolus. Then surrounding it we have nucleoplasm, and then surrounding that we have a nuclear membrane which has nuclear pores. You need to be able to know how to draw it. And also, learners confuse nuclear membrane with nucleoplasm and nucleolus with nucleus. Please make sure that you understand the different terms and you know how to label the different structures on the diagrams. In the nucleus of almost every cell in your body is the collection of DNA needed to make you. Now let's just back it up a little bit. In grade 10 as well, in chemistry of life, you learned about DNA. Okay, and you learned that it's called deoxyribonucleic acid, and you learned basically that it's a nucleic acid and it is found inside the nucleus. So this is reminding you now from your grade 10 that the DNA is found in the nucleus, and it's a collection that makes up you, that makes you. Okay, it's needed to make up you, your genetic makeup. It makes you who you are. On each chromosome, genes, which are segments of DNA, code for all your characteristics. So everything that makes you who you are, it all comes from your DNA, because those segments of DNA, which are called genes, those are what make you, make the characteristics that you have. Then we go into the brief history of DNA quickly. You need to know who these people are, and you need to know what they were responsible for. So in 1953, we have James D. Watson and Francis Crick and Maurice Wilkins. They devised a double helix model for the DNA structure. You need to know that it's a double helix. That is the shape. If you wake up in the middle of the night and I have to ask you what is the shape of DNA, you need to say it's a double helix. Their breakthrough was made possible by the work of Rosalind Franklin, whose X-ray diffraction studies of the DNA molecule shed light on its helical structure. Okay, so Rosalind Franklin was the one who did the X-ray diffraction studies, and then she also contributed to seeing this helical shape. But it was Watson and Crick and Wilkins. They devised a model to show that it is the double helix. Okay, so you need to know that, and you need to know the differences between what they discovered. Now we're looking into the DNA structure. As we said, it is a double helix. You cannot say it is a helix. You cannot say it's spiral shaped. You cannot say it's double stranded. You need to say it is a double helix. Okay, in life sciences, it is very important that we are specific, especially in a topic like DNA, genetics, meiosis. In most of our topics in life sciences, you need to be as specific as possible. So in this one, we can see over here, this is the double helix that I'm talking about. Can you see that? So yes, it is two-stranded. There are two strands, but the shape is a double helix. The DNA consists of two strands that are arranged into a ladder-like structure called a double helix. So you can see in this diagram over here that this is a ladder. Okay, so you have, so these structures over here are the rungs of the ladder, okay, where you would step your feet, right, to climb the ladder. A molecule of DNA is made up of millions of tiny subunit monomers called nucleotides. Okay, so we have this big double strand, and then each strand, this strand is made up of monomers, which is your simple units. Okay, so what is the most simple unit of a nucleic acid? It is a nucleotide. So this structure over here is a nucleotide. It is made up of a phosphate group, okay, which is this black dot, a pentose sugar, which in this instance is a deoxyribose. You can see there it is 5-sided. That's where it gets its name from, pentose, okay, and its name is deoxyribose. You need to know that, not deoxybose. Sometimes learners leave out the RI, or sometimes they leave out the O. Okay, you please spell it correctly. It is deoxyribose. Okay, or sometimes they say deoxyribonucleic acid, but the sugar is deoxyribose. Then we have the nitrogenous bases. We have adenine, thymine, guanine, and cytosine. Okay, so it's important that we know these nitrogenous bases, and we know what a nucleotide is made up of. They can ask you to draw it. Okay, they can ask you to label it. So if you get this DNA structure, you would know how to label it. Okay, now we move on to the next slide. Or before we go that, I just want to show you that what's also important to understand here is that you can see there are two strands, right? Okay, so this is strand number 1, this is strand number 2. So you can see that these bases, these nitrogenous bases, they are paired in a specific way. So you can see from the shapes that they actually join together, or that they complement each other. That's what we call it. We say that they complement each other. So how it works is that we have adenine that it pairs with thymine, and we have guanine that pairs with cytosine. So you need to know that, because we cannot have a situation where we have adenine pairing with cytosine or guanine, and that's not the way that the bases are paired. This is important for when we do protein synthesis as well as DNA replication. Right, then we have RNA. RNA is a little bit different. You can see from the structure as well, it's single-stranded. It's a single-stranded molecule, and it's also made up of nucleotides. But these nucleotides are different. So here we have a nucleotide as well. It's not like a ladder. It doesn't have two strands. It's only got one strand, and it's made up of a phosphate, a pentose sugar, as well as a nitrogenous base. Okay, so once again, they are adenine, guanine, and cytosine. But the difference is, can you see what the difference is? Give you half a second. The difference here is with number 2, where here we have uracil, whereas we normally have thymine in the DNA. So that's very important to know, and you need to know the structure of DNA and RNA very well in order to understand what we're going to do next, which is your DNA replication, and then protein synthesis as well. It's for later on. There are 3 types of RNA. You need to know them. There's messenger RNA, there's transfer RNA, or ribosomal RNA, and ribosomal RNA. These terms in brackets are accepted. You can use them: small letter M RNA, small letter T RNA, and small letter R RNA. Okay. We will go into a little bit later on the functions of those. Right, so this is showing you a picture of a nucleotide. As I said, you must be able to know how to draw this. It's a nice question that they like to ask. And then you need to know that what the nitrogen bases are. They are guanine, cytosine, adenine, and thymine in DNA. And in RNA, they are guanine, cytosine, adenine, and uracil. Right, now we come on to the process of DNA replication. So as I said, DNA needs to replicate. It's a very important process that forms part of cell division. That is mitosis and meiosis. Okay, so let me just write it here to remind us that cell division, there's mitosis. Mitosis, we have learned from grade 10. I'm just going to put it there in brackets so you can remind yourself. And then this year we are going to be learning about meiosis. Okay, so the process, you need to know this process off by heart. You need to know how to describe the process. It was actually asked in last year's paper. We're going to look at that question that they asked on DNA replication, but let's go through the process. So first we have the double helix unwinding. So remember we said that it is a helical structure and it's bound up. Okay, so it first needs to unwind, and when it unwinds, it's going to now be, it's going to look like the ladder, right? And then what happens is the weak hydrogen bonds between the nitrogen bases break, and the 2 DNA strands unzip. I just want to go back to show you that quickly. I didn't mention that. These little dots here, those are the hydrogen bonds. Okay, so your nitrogenous bases are held together by hydrogen bonds, and they are weak hydrogen bonds because they actually break when after it unwinds during DNA replication. So the double helix unwinds, the weak hydrogen bonds between the nitrogenous bases break, and the DNA strand unzips, or it separates. Okay, so that's what causes us to unzip. Now it's very important that point 1 and 2 you don't mix up. Let's think about it logically, right? Here it's unwound, okay, and now we need to it's wound, sorry, we need to unwind it, okay, and it needs to unzip. It cannot unzip until it is unwound, because when it is like this, right, you can try zipping it. You can actually use a zip and try it for yourself. Twist the zip. You won't be able to unzip that. You won't be able to move the zip while it is twisted. But once you untwist it and you unwind it, that's when you'll be able to zip, okay, to unzip. So it's very important that you don't confuse those two. You will lose marks if you do that. Each original DNA strand serves as a template on which its complement is built. 3 DNA nucleotides build a DNA strand onto each of the original 2 DNA strands by attaching to their complementary nitrogenous bases, A to T and C to G. This results in 2 identical DNA molecules. Each molecule consists of 1 original strand and 1 new strand. Okay, so let's look at the picture. We can see here that it is wound up at this point. Then it unwinds. There's point 1. Then it unzips. So you can see there it unwinds, it unzips because the hydrogen bonds are breaking. Now we've got the template. There's a template here and there's a template here. So can you see there that these nitrogen bases are now exposed? Can you see that? They're exposed, yeah. At this point, they're exposed. So now we have your free-floating nucleotides. They come from the nucleoplasm. Okay, so remember we are in the nucleus now, right? And then these are in the nucleoplasm. We have our nucleotides that are floating around. So now they are going to complement the strand. So we're going to have C complementing with G, T with A with T, until we are going to form the strand, right? And the other side, it's going to be the same. So then it's going to wind up again. So in the end, we are going to get 2 identical DNA molecules. Okay, and where learners go wrong here, they go wrong in a few places. Number 1, they don't talk about the double helix unwinding. They just say DNA unwinds. Another place is that they say that it unzips before saying unwind. They talk about 2 in bullet number 5. They write about 2 identical DNA strands instead of DNA molecules. Okay, so the molecule is made up of 2 strands. Okay, but it's not 2 identical strands that are formed. There are 2 DNA molecules that are formed. Okay, so that is what you need to understand and you need to remember, because when you write them in the exam and you're not specific and you don't write the you're going to call it a DNA, 2 identical DNA strands, you will lose marks because now you are changing the meaning of what is actually happening. Right? Then another point, where does it take place? It takes place in the nucleus of the cell. And when does it take place? It's during interphase of meiosis and mitosis. Very, very important for you to know this. You need to know that it takes place in the nucleus, and you need to know that it takes place during mitosis and meiosis. What is the importance of this process? Remember, I told you it's part of the examination guideline that you need to know. What's the importance? The importance, number 1, is cell division. I'm reminding you again, what are the 2 cell divisions? Mitosis and meiosis. What's the other importance? It allows each chromosome to be copied so that each new identical daughter cell produced contains the same number and type of chromosomes. Okay, so it allows each chromosome to be copied so that when cell division occurs, each new identical daughter cell is going to contain the same number of and type of chromosomes. Right, so here we go, DNA replication again. It's just giving us a different diagram. I love this diagram because you can see it in more detail what I was talking about. It unwinds, the weak hydrogen bonds break, it unzips, and then you can see over here that each DNA strand serves as a template. So here we can talk about strands because this is not a molecule and this is not a molecule. These are strands, okay? But each strand is serving as a template where the complement is going to be built. So these are, can you see they have broken here? So now there's a space here. This space is going to be filled now with the complementary strand, nucleotides. So then we have the free DNA nucleotides. They build a DNA strand onto each of the original 2 DNA strands by attaching to their complementary nitrogenous bases. And then they're showing us here that they are the free floating nucleotides, the DNA nucleotides from the nucleoplasm. This results in 2 identical DNA molecules. Each molecule consists of 1 original strand and 1 new strand. So you can see here, this is molecule 1 and this is molecule 2, and you can see that they are identical. How do we see that? Because of the arrangement and the sequence of the nitrogenous bases. Okay, that's what you need to understand. You need to understand that we had a DNA structure that had nitrogenous bases that were paired together. They were paired in a particular sequence, because that particular sequence is going to be a gene that is going to code for a specific characteristic. It's in a particular sequence. So when it replicates, it needs to be an exact copy. And how do we see that exact copy? We look at the nitrogenous bases because they need to be identical. That's how we know that DNA replication has taken place. Right, let's go through a question. Okay, I'm going to give you about 3 minutes to go through to do this question. Okay. You can skip number 1, you can do number 2, 3, 4, and 5, and I'll give you 3 minutes. Welcome back. I hope that was easy for you to complete. Let's have a look at the answers. So question 1, the answers were here. That's why I said let's do it together. So the first part is that they're asking us to identify certain structures. But before we go into that, I cannot leave here without actually explaining to you how do we go about answering questions. So we need to first always analyze the diagram. What is given to us here? They're telling us, because remember we're doing a small section now, but when you're going to get the paper at the end of the year, it's going to be mixed up with everything else. And out of all of the diagrams that you need to learn, you need to focus and see what is this diagram telling me. So the question says the diagram below represents part of a DNA molecule. So they're nice enough to tell us that it's DNA. Okay, they could have just said it's a nucleic acid and you would have had to think for yourself that it is DNA. But let's just think about it quickly. If they had said the diagram below represents part of a nucleic acid, and you got this diagram and they don't tell you that it's DNA, what would be the indicators to you that this is DNA? So firstly, it is double-stranded. Okay, that's the main thing that you can see here, that it is double-stranded. Okay, there's no other indication here. If the nitrogenous bases were labeled and there was thymine, then you would also be able to say that now this is DNA, because you can see that there is thymine and not uracil. Okay, so we're going to identify molecule X. So we go to X. What is X? It is a nitrogenous base. You cannot be specific as to which nitrogenous base it is because there's no indication in the question which nitrogenous base it is. That being said, it's very important that you listen to what I said there, because in some of your textbooks, they actually show you these shapes, and they show you that this shape is adenine and that shape is thymine, but it isn't so. I really want you to take note of that, because some of the textbooks have it like that, and then the learners think that those are the actual shapes. Those are not the actual shapes, it's just a representation. So you cannot learn it like that. The only way for you to know is that if it was labeled here, say for example, this is cytosine and this is guanine, and then this is adenine, then you would know that that is thymine because of the pairing, not because of the shapes. Okay. Then sugar at Y. So we look at Y. What is that sugar? It is deoxyribose. Okay. You wouldn't be able to say pentose sugar here because you need to be specific. Remember, this is DNA. There's also a pentose sugar in RNA. It's just that it's called ribose. And then we have the bond at W. The bond is an hydrogen bond. Okay. Give the collective name of parts X, Y, and Z. So collectively, X, Y, and Z, what are they called? It's a nucleotide. State the natural shape of the DNA molecule. The natural shape, it is a double helix. If you said helix, it is wrong. If you said double-stranded, it is wrong. If you said that it's a double-stranded structure, it is wrong. Okay, it's a double helix. Name the process whereby DNA makes a copy of itself. Right? That is DNA replication. And name 2 places in an animal cell where DNA is located. It's located in the mitochondria as well as the nucleus. Okay, I didn't mention it earlier, but it is something that you need to know, that DNA is located in the mitochondria and the nucleus, and it's actually also located in the, oh, they're asking for the animal cell here. So you can only say mitochondria and nucleus, but it's also in the chloroplast, but that is only in plants. Okay, so that's why it's important to remember grade 10. Because now, if you had just learned this from the examination guideline, and some of you might have answered chloroplast here, and you wouldn't get it right, because this question is asking you for the animal cell, where DNA is found in an animal cell. Okay, then we have describe the process of DNA replication. Right, now this one I also want you to do. I'm going to give you another 3 minutes. DNA replication. You need to describe the process of DNA replication. Welcome back. So let's look at this question again. Describe the process of DNA replication. Now, when they ask you to describe something, you just need to jot down all the facts that in a logical sequence of what happens in that particular process. Okay, I'll say that again. You write down all the facts in the correct sequence of the particular process which is being asked. Okay, so we start with number 1. What did we say number 1 is? Hopefully you got it right. The DNA double helix unwinds. Not the DNA strand unwinds. Forget about the word strand when you're talking about DNA. Okay? But when you're talking about DNA, the DNA structure itself. The only time we are using the strands is when we're talking about the template. We are talking about they're separating into 2 strands. We have 2 separate strands. That is when we are talking about strands. Otherwise, the DNA itself is a molecule. Okay? So the DNA double helix unwinds and it unzips. Or you can say the hydrogen bonds break. There is 1 mark for saying unzips or the hydrogen bonds break. You don't get 2 marks for that. To form 2 separate strands. Both DNA strands serves as a template to build a complementary DNA strand, or you can say A pairs with T and C pairs with G. Using free DNA nucleotides from the nucleoplasm, this results in 2 identical DNA molecules. Now, in this particular one, this was actually taken from last year's question paper at the end of the year. So in this particular one, there were 1, 2, 3, 4, 5, 6, 7 possible marks. Okay, and the ticks are in a particular place, right? But it doesn't necessarily mean that it's always like this. Okay, so it's important that we understand when we go through past papers and when we go through previous question papers that we don't just learn the marking guidelines off by heart. Okay, and we don't learn questions off by heart, because they will change. Every year they will change. It's very seldom that you're going to get the exact same question again. Or the marking guideline might be different. So sometimes they might give you a mark for saying nucleoplasm. Sometimes they want you to say the hydrogen bonds break. Other times they don't give you a mark for saying, they could be putting the DNA in brackets where you only have to say the double helix unwinds. Other times they would want you to say DNA. So learn the processes off by heart, but learn them properly. Okay. Right, now we're going to move on to DNA profiling. I'm going to hand over to my colleague, Nonhlanhla Mshali. Stay tuned and enjoy.
grade 12 learners. Welcome to the second part of the Life Sciences lesson. Let me remind you again of our WhatsApp number so that if you have questions, you can contact us and we'll do our best to answer your questions. The WhatsApp number is 079-454-9535. I'm going to repeat the number again. It's 079-454-9535. So the second part of the lesson is mainly on DNA profiling, as you can see on your screen. So what is DNA profiling? DNA profiling is a technique that is used to identify the sequence of bases. Earlier, you learned about the structure of the DNA and the nitrogenous bases that make up the DNA. So here we're looking at the sequence to identify the bases, and that technique to identify the sequence of bases in the DNA molecule is called the DNA profiling. And the DNA profile itself is just a pattern of lines, as you can see here, a pattern of lines that are different in length, that are different in sizes, and some of them are dark and some of them are lighter than the others. So this, what you see here, is what we call a DNA profile, this pattern that appears here. And then the process of getting this pattern is called DNA profiling. So what happens during DNA profiling? The nucleotides are separated from each other in the order that they are found in the DNA strands, and then these nucleotides are the ones that appear as dark bands. Some may say they appear as what? As lines, because what you see there, it's different shades of bands or different shades and sizes of the lines. So DNA profiling, what you need to know is that DNA profiling is not the same as fingerprints. Your fingerprints is what you see here on your hands. That is completely different from what we are talking about here, which is DNA profiling. So do not confuse the two. In actual fact, you are not even supposed to be taught about DNA fingerprints or fingerprints. It's just DNA profiling and the DNA profile. And then, why DNA profiling? Remember, there are a lot of things that go on in the world. There's crime, and you need to be able to identify who are the crime suspects, or you need to know who the father of the child is, or we need to find matching tissues for organ donation, for example. So to be able to do that, scientists need to analyze the DNA profiles of different individuals so that at the end you are able to check whether they match, and if they match, then that person can be able to donate, or we can police or whoever is involved can easily identify who the crime suspects. So what is involved when you analyze the DNA profile? So what we have here in front of us is an example of the DNA profile, and as you can see, I said it is different lines of different sizes. Sometimes even the thickness is not the same, but the one that is in front of us has the same thickness, and the size of the lines is also the same. So here we have profiles of 6 different people, and they are represented as what? As the dark bands. So when you look at this, you can see that there's 2 people that are related here, and those 2 people that are related are twins. So one thing about the twins that you need to know is that each and every person has a unique DNA profile, except for for the twins. So those are the only people that have the identical DNA profile. The rest of us have a unique DNA profile that does not match anyone else. So here, when you study the DNA profile, so when you analyze, let's say we are analyzing DNA profile, what is it that you need as a grade 12 learner? You need a ruler, of course, because what are you going to do? You are going to look at the lines. Remember, we said we're looking at the size of the line, we're looking at the length of the line, we're also looking at the thickness of the line. We are also looking at the position. So are they in the same position? Are they of the same size? Are they of the equal length? And then if they are like that and they are found in the same positions, and if you are matching 2 samples, for example, let's say you are given 2 samples, the one that is found in the evidence and the one that is unknown. Now you must check whether these 2 things match. So that is what you're going to look into. And then at the end of the day, if you find that on the unknown sample, most of the bands match the one that was found as evidence, then you can conclude that this 2 belongs to the same person, so they match. So it's either all of them will match or most of the bands are going to match each other. Then your conclusion will be, then these are what are identical or they belong to the same person. So in this DNA profile that we have here, we have 6 people with their DNA profiles, and we need to identify the 2 that are twins. So you need your ruler. When you take your ruler, you're going to put it on the first band, for example, and then you're going to see whether how many people have, or in this profile, how many have their DNA brand, the first one as number 1 there. So if you look into this, the first, this A, B, and D, their bands match, but you cannot draw conclusion yet. And then if you go to the second line, your ruler will be placed like this, and you go further down, again, you still have A, B, and D that are matching. If you go on to the third one is only C, E, and F. And then moving down again, it is only B, C, E, and F. So this one is not found in the other ones. But then if you look at C and D, already if you look at C, let me see. If you look at C and F, you can see that this band here is exactly the same as this band here over F. Again, if you look at this one, the here and there, they match. Also, they match with what? With E. So E also has this, they have the same bands in the same positions. And then if you move further down with your ruler, that's what you're going to do. This band is only found in A and nowhere else. The band number 4, it is found on A, it is found to person C, person D, and person F. So again, we have C that has the band, and then we have this one, and we have what? We have F. So if you look, you can already see that there's 2 people that look like they have the same bands in the same positions, and that is your C and F. But let's continue to look at the others, the rest of the bands. With this one, it's only B and E. And then we have this one, which is only found on person D and nowhere else. Then if you go further down, it is only person A and person B. If you go further down again, we have C and E and what? And F. Again, C matches E and matches what? F. And then if you look at C and F, they already have 4 bands that are matching and they are in the same positions. Okay, going further down, again, we have A, but we also have C, and we also have what? Have F over there. And then going further down, this one is only found in B and C. And then this one is only found in A, B, D, and E. And then the last one, it is only found in person C and person F. So here, we can then conclude that the people or the twins in this DNA profile are person C and person F, because the bands that they have are found in the exact same location and they've got the same number of bands. So then we can conclude that the 2 that are twins is person C and person F, because their bands match. So that is how you're going to study the DNA profile. You need your ruler. You cannot just eyeball. I was using a pen drawing because I don't have a ruler here with me, but with you, when you're getting a task or you're writing an exam or anything, when you do this activity, you need a ruler so that you put it down and you draw these lines so that you identify which bands match and which bands do not match. Okay, then here we have an activity now on what DNA profiling looks like. So here we have question 1 and question 2. I'm only going to give you 2 minutes because there's a diagram that must be studied and then there's 2 questions that must be answered. So let's give you 2 minutes before we look into the answers. Okay, your 2 minutes is up. Let us look at the answers together. So here the question is, the diagram below represents the DNA profiles of 3 children. So we have 3 children and their parents. Only 2 of the children are their biological children and 1 is adopted. So we have 2 that are biological and 1 that is adopted. Like Vedika said earlier, when you are given a question, life sciences paper has many diagrams. So you must be able to study. When the question says study the diagram and answer the questions that follow, you must study the diagram. So before you even go to the questions, you need to make sense of what is going on here in this diagram before we go on to the questions. So what we know is that we have 5 profiles, DNA profiles. We have 3 children, Uhela, Priya, and Leo. And then we have the parents, which is mother and father over here. And then what we are told is that 2 of these children are biological and 1 is adopted. So then the first question says, identify the 2 biological children. So what are we going to do? Again, you said you take your ruler and you put it there, then you try and find the bands that are in the exact same location. So if you look at mother, has first band over there, and this band is also found on child number 1, which is Hela. So Hela matches mother over there, and Priya and Leo do not have. And then if you go to the second one, this band from the father matches what is found on Hela and also what is found on Leah. Because if you look carefully, this one on Priya's profile, it is not exactly on the same location as this. It is slightly above. That is why you would need your ruler. So it is somewhere in between this and that one, but it's not in line. So again, here we have this band that is found on father, which is also found on Hela and also found where on Leo. Then if we go further down, then we have this one, and this one also is not in line with any. But then we have this, which is in line with the one that is found on Leo. So Leo has a band and matches who? The mother. And band number 4 from the mother. Okay, this one does, okay, no, it is also slightly above this one. If you put a ruler, you see that this one is not in line with this. And then we have band number 2 from father. If you go further down, it does not really match anyone. And then this one also does not really match anyone. And then we have this one from father, which matches the one that is found on Hela and matches that is what the one that is found on Leo. So we have this and this that match. So already you can see, without even going to do the last one, you can see that most of the bands on profile Yaga Hela and Leo's profile contains the bands that are found where in the DNA profile of what of mother and father. So we can conclude here and say the 2 biological children here are Hela and Leo, because most of their profiles or the DNA, the bands that they have are the bands that are also found in the DNA profile of what? Of the mother and father. So that is why, explain your answer in question 1. The reason why we are saying it is Hela and Leo, it is because most of the bands in the DNA profile of Hela and in the DNA profile of Leo match the bands that are found where? In mother and father. So let's see. The first answer, we said identify the 2 biological children, and those children are Hela and Leo. And the answer, explain your answer. It says all of the DNA bands from Hela and Leo match with the DNA bands of the mother and the father. So the last one that we did not do, if you see, it also matches. It is found here. And then this one of the father is also found here. So all the bands that they have, they match the bands that are found on the mother and on the father. Or you could have said none of the bands, because none of the bands on Priya matches the one that are found in the mother's profile or the father's profile. So none of the bands from Priya match with the DNA bands of the mother and the father. So this is how you respond. you cannot just say the bands or the bands inhaler match the ones that are found in mother. You either say all of them or you say most of them, depending on the number of bands that match when you respond. And in your answer, you must include mother and father, or you say the parents. That is also important. That is how the marks were allocated. And then here it's still the same question which you're going to do together. The diagram below represents again DNA profile of 3 children. So it's a continuation of the first part. Here it says state 3 other uses of DNA profiling. So here what we're looking at was paternity testing, who the children belong to, or who's the mother and who's the father, or who are the children. So that was paternity testing. But what are the other uses of DNA profiling? I did state that when I started that it is also used in identifying what the crime suspect during investigations. So that is one other use of the DNA profile. You also know about organ donation, right? So you cannot just donate organs. There's processes involved when organ donation is going to take place. The tissues must be compatible. The tissues of the donor and the tissues of the person that is going to receive, those must be compatible. So how do scientists look into? They look into the DNA profiles for compatibility. And the other thing is to identify dead bodies. If there's someone that is dead and they don't know where they belong, they can do the DNA profiling and be able to identify who those people are. And the other thing is to find, we say that lost relatives, say of relatives that you are looking for, but you are not sure whether they are your relatives or not. You can also do DNA profile to determine whether you are related to those people or not. So there are many other uses of DNA profiling out there, and it is important that you know all of them. So here what you are given is tracing of the missing person, identification of the genetic disorders. It's one that I did not mention. Yes, identification of the genetic disorders that are not known, and scientists, they make use of DNA profiling to study those to identify what they are. Identification of the suspects in a crime, matching tissues for organ transplant, organ donation, and identifying dead persons. So these are all the uses of DNA profiling. So the only one that is missing here is the one that was done in the diagram, which is the paternity testing. And the questions around this, they always come out like this. The question will be, what are the other uses of DNA profiling other than the one that is shown in the diagram? And you will see as you practice, because most of the past question papers, there is one question on DNA profiling. And the more you practice, you will see that the answers are more or less the same. But you will have the DNA profiles that you will get are not the same. So that is why you need to practice more, use your ruler, and see how the answers are given in different question papers. Then you will have enough confidence to answer any other question that come on DNA profiling. And then the last question here says, explain 2 reasons why sometimes DNA profiling can prove to be controversial, cause people to disagree with the results. So why would this prove to be controversial? Number one, let's say in a crime situation, for example, a sample can be planted in the crime scene. You find that it does not actually belong to that person was not even at the crime scene, but the sample of their blood or saliva or anything was planted there. And then if it proves that it is that person, then people can be arrested when they actually did not do anything. So that is one of the reasons why. That is one of the reasons why it proves to be controversial, because samples can be planted and it might put people in jail when they were not even supposed to go there. But let's look at the answer. So what we have here, DNA samples may be planted, a person can be framed with use of false evidence. 2, human error, which can lead to false results. A small amount of DNA is analyzed, therefore it may not necessarily be the DNA profile of that individual. And also it is done in private labs, so testing standard may not be followed because there are many labs out there where DNA profiling can be done. And invasion of privacy, that's another reason why it is proved to be controversial. because what are we doing? They are revealing personal information. What if you don't want your personal information to be revealed? So that is why again we say it can prove to be controversial. Sometimes other people will tend to disagree with the results, and it's because of this, that the sample may be planted, there might be human error, there's lots of private labs out there, so there's not a set procedure that is followed. And lastly, we're looking at the invasion of privacy. And then we have come to the end of today's lesson. Next week, we're going to continue with RNA and the different types of RNA, protein synthesis, and mutation. So please tune in our next week's lesson, which is also on Thursday at 2 o'clock. We will see you there.